So I wear a hat as both seeing adult and children with rheumatic disease, including lupus. And I want to share with you some of what I think is really important to understand about the past 2 decades of lupus research and paradigms that have really changed how we think about lupus. And I got to thinking about how to deliver this, so you'll let me know if it's successful, because I now have a niece who just defended her PhD at Northwestern in communications, and she's really, her area of focus is communicating science to the public. So some of the stuff is going to be complex, and we're going to focus on what's important for us to know, so don't get intimidated by any of the slides. I also wear a hat in the research division and in the CASE Award as Hub Research Capacity for the Clinical Translational Science Award at CASE. Okay. How do we? Okay. These are my disclosures. We are at Metro a member of something called the Lucid Network, which is an academic clinical trials network. And I have a current trial using tofacitinib to treat cutaneous lupus that was initiated by Hermine Brunner, and we're her second site. And I'll tell you a little bit more about that just as a plug, because if you know anyone, send them our way. And I was a co-investigator with Dr. Ballou, being the lead site PI for another company. So when I was very early in my career, I remember very vividly that there's this woman, Hallie Matzinger, who was an immune researcher at NIAID, and she did this and shocked people in the audience because she talked about danger. And so I think that's actually one of the real messages that the innate immune system, or the immune system that we all have that defends against danger, is really important in lupus. And so we talk about having an innate immune system and an adaptive immune system. And so the innate immune system is what's responsible for putting out that fire if you get called, if a fire engine gets called to a fire. And then the other piece is the adaptive immune system, which is really responsible for maintaining quiet in the immune system. And so what from early discoveries really by Peggy Crow's lab at Special Surgery and also Virginia Pasquale, who was then in Dallas, and Lynn Punaro, who was her clinician partner, who looked at pediatric lupus. It's become incredibly important that type 1 interferons, or something called interferon alpha, is really important in lupus. It wasn't completely understood how that worked, but we all know very well that patients with lupus get a variety of autoantibodies. And so when I graduated fellowship in 1994, we had a poor understanding about how did antibodies get into cells to make it to the nucleus, which has its own membrane and walls. How did they get into the nucleus to make antibodies against this double-stranded DNA and other things that were contained there? And so Mariana Kaplan, who was at Michigan, who was 2 years behind me in fellowship, and is a good friend, really began the neutrophil netting story, and I'll tell you some about that. And then you can always, it's sort of like if you think of innate immunity as the west side and adaptive immunity at the east side, you all know if you're from Cleveland, people don't want to cross it. I say that because I say that I'm allowed to cross the Cuyahoga every day because I'm not from Cleveland. And so the dendritic cells really bridge that crosstalk between innate immunity and adaptive immunity, or they're the people who are willing to drive the Valley View Bridge back and forth. And so we know, and I'll show you some of this in the context of another disease as well, that the factors that initiate and perpetuate inflammation are different. So what starts the process isn't necessarily the thing that fuels the process for years and years. And so those are different concepts. And genetics I'm not really going to address today, but we know that there are missing genes from people or monogenic disease where one gene coding for a protein is bad, and that fuels lupus. But if you look at lupus in the general population and not families of lupus or early onset lupus, you have multiple genes that contribute. And it turns out that those genes also contribute to autoimmunity in rheumatoid arthritis and thyroid disease and insulin-dependent diabetes. And so what you often find is not necessarily a family history of lupus, although some people have that, but it might be a mother with rheumatoid arthritis and an aunt with psoriasis or other autoimmune features. And so the type 1 interferons are really recognized as playing a critical role. And so we know that in lupus patients, but not all lupus patients, if you measure alpha interferon in the blood, that it's elevated during flares. And that's incredibly difficult, actually, to measure the protein in the blood. It's much more easier to measure the genes that get turned on by it. It's just that the assays are easier. And so typically when we look for alpha interferon activity, we may not be measuring plasma or blood alpha interferon, but instead we're looking at the DNA and RNA, and mostly the RNA. So the DNA is the gene coding. The protein is what you can measure in the blood, just like any other protein you might consume. And the RNA is what bridges the DNA code to the protein. So RNA is often what we measure because we're looking at the genes that code for the protein and whether or not they're up, because often, but not always, the protein levels follow the genes, and it's often easier to measure what we call RNA or gene expression. And so you'll see some gene expression tests rather than direct protein level measurement, because direct protein level measurement for interferon alpha can be really challenging. And you want something in research that's reproducible. It is quoted. This is Virginia Pasquale and Lynn Punara's data from the Journal of Experimental Medicine about 16 years ago. And what they did is they looked at children with lupus because it was felt if you have early onset, you're more likely to have genetic causes of it. And it was because lupus patients accumulate damage over time, that if you study children, that you wouldn't have a lot of coronary artery disease, heart attacks, other kinds of things that play into how your genes change as you age. And so what they did is they looked at a kind of adaptive immune cell called monocyte, and that's shown right here. And they looked at its activation by lupus serum, shown in the next panel, and you sort of see these cell things spreading out, and that's really for communication with other cells. And in the top panels, what you see is that when they added the serum, so the blood minus the red cells, to monocytes, that these monocytes were able to stimulate T cells. So you should take away a couple of different things from this. One is there's incredible crosstalk between the cells in your blood. And that's why often blocking one thing might work for a while, but then the immune system becomes resistant to it, or you need a two-pronged approach or a three-pronged approach. And they then showed, so the pink, the blue, and the yellow are serums from different patients, as is the one, one is a normal donor and one is lupus on the bottom. So the patient that goes along with the normal didn't appear to have a lot of whatever substance was in the rest of the lupus patients. And as you know, I don't have to tell you that lupus is heterogeneous, right? So some people get mostly skin lupus, some people get mostly arthritis, and some people get kidney disease and nothing else. And so really lupus is probably a group of several, if not many, disorders, and that's been often the problem in studying it in clinical trials. And you'll look when I show you some inclusion-exclusion criteria about what the problems are. Because in clinical trials, we want one single outcome measure that will tell us whether or not the agent worked ideally. And if you have lots of them, it's really hard to sometimes meet the bar of getting something to market or knowing for sure if it works. Okay, so this is what I was talking about gene expression, and all I want you to understand is these are normals, and there's a little bit of red and a little bit of blue. So red's up, blue's down. In the middle, this is the SLEDAI, so the SLE disease activity score. And so this is less active patients, and this is more active patients, and this is highly active patients. And you can see that this looks a little different from this, but this looks a lot different from this, and this looks even more different. And they don't look very much like the arthritis patients. And so that's a clue that there are different genes involved in the inflammatory phase based on how active the lupus is. Okay. So I talked about the innate and the adaptive immune system. Is this a pointer here? Yeah. Okay. Is there? Okay. So if we think about virus as what is the danger, right? It's been all over the newspapers that virus is danger. And we think about the dendritic cells eventually taking that up, talking to B cells and T cells, and interferon alpha is important in this, and then it secretes things to mature B cells. So B cells that produce antibody are called plasmablasts, and the things that we think are these plasma cells or long-living plasmablasts in lupus patients are stuck in the tissue, not in the blood, much harder to get to, but they are probably largely responsible for continued production of antibodies against yourself in lupus. And so how do you deal with this? Whoops. So what we didn't know very well is how do you get from here to here? Is there a light we can just turn down for a moment? So this is an early diagram from Mariana Kaplan's group. And what you see here is you're looking at the blue are the nuclei of cells, and the red, I believe, was one of the, I think it was elastase stain or something like it, because neutrophils contain something that is a digesting enzyme. So think about this was control serum, and this was lupus serum, which had something that was digesting and allowing neutrophils to kill themselves and degranulate themselves and sort of, if you think of an open stomach wound, the neutrophil is dumping its contents. And so what Kaplan was able to show is that control serum didn't induce most of this neutrophil netting, but lupus serum did. And when you look at where it is, you can see that the contents in blue are being dumped into these red things attached to the neutrophils. And so the neutrophils, you can turn it back on, have these wide nets, and they've taken everything in the nucleus, and they're saying, here I am, and they're in the blood. And that's one of the things that happens in lupus to get the immune system revved up. And so hence the mystery of how are these things getting out of the cell for us to make antibodies against them. Because they were in a protected ball. You can think of lots of different things that are in a protected case. If you have a balloon, the air is protected by the outer of the balloon. If you pop it, the air goes out, and if it's helium, it spews all around. And so that's in part how, when you're challenged with infections or other substances, the nuclear contents get dumped and are available for B cells to make antibodies against. Okay, so these are some of the antibodies that have been identified at that point on these neutrophils. And some of you will recognize things like C1q, which is often up in lupus, or autoantibodies are there. But there's just everything in that cell is capable of getting spewed. So what happens is that this neutrophil is here, and as the nucleus condenses, then the plasma membrane ruptures, just like your balloon. There's net formation, and all of those proteins that are now available to the immune system to make antibodies against rupture. And they can do it one of two ways. They can either do it by balling up all their DNA products in the nucleus and then dumping them out, or by just dumping out without balling them first. And that's probably not as important for us to understand today. So we know viruses stimulate nets, but not every lupus patient has a virus before they get inflamed. And so that we know that there are other things. There's DAMPs, or danger molecules. There are fungi that can activate this immune system. And gout and pseudo-gout can also. So this is a common mechanism in diseases of what we think about as innate immunity. And even though it was described early in lupus, there are multiple other diseases that use this process, including gout and some of the inherited diseases. So this is a little bit of a sloppy slide. It's sort of an alphabet soup. I didn't put every company up there. So I sat last night and said, because Adrian, I think, had asked, what are the clinical trials that are going on? What are the targets? So there are about 365 trials listed in the United States in lupus. Less than 200 are interventional. And there's almost 200 that are either actively enrolling or have enrolled, and the trial is ongoing without results. And so there are things that are coming to antibodies against TYK2, low-dose interleukin-2, which you'll hear about. Not only is there Janus kinase inhibitors like Xeljanz, which we use in RA, or tofacitinib is its generic name. There's anti-CD6 that's near and dear to my heart, though I think there may be a better antibody coming along that I've worked with for this. This is going into trials for lupus nephritis. It's been through its normal human testing. Anti-CD40 has been a target. There are multiple companies that have targets. Novartis does Boehringer Ingelheim. There was initially not very much enthusiasm because about 15 years ago there was a trial of an anti-CD40 antibody that was associated with clotting. And so you got to figure out what part of it is causing the clotting and what part of it is useful therapeutically, because you don't want to have you want it to be therapeutic, but you don't want your window to be so small that you have to be right in a narrow therapeutic window, because that opens it up for more side effects if the therapeutic window, as we call it, is very narrow. And there's belimumab has already, there are some phase 4 trials. So what did all these companies have to go through? And I was asked to speak a little bit about stem cell therapy. And I'll tell you a little bit about the RA work. RA is a little bit easier in some ways because even though it's heterogeneous, it's not as heterogeneous as lupus is. And if you can do a human study in RA and you know it's safe, then it will also be an easier road potentially with the FDA because the FDA decides what outcome measures can be used to market a drug, essentially. So you have to meet some bar for each disease. And we had some preliminary data in RA. So for an investigational new drug to come to market, you need to first have a concept and some premise of why this thing might work. You need mostly first in human, usually require animal preliminary data or other preliminary data. And there are extensive sections in an investigational new drug application of chemistry, manufacturing, and controls. So these are some of the things we've seen come and die, come and stay and come and die. There are antibodies to anti-CD20, and there are other ones in process. There's anti-IL-10, which had not gone forward, anti-CD22, which has not gone forward. CTLA-4 Ig is abatacept. There are studies in lupus. It's used routinely. This apoptotic material is dead cell. So instead of calling it apoptotic material, we could just say dead cell material. This is B cells, and anti-BLyS or anti-BAFF, which is a B cell thing, has come to market. It's known as belimumab. So some of you know Benlysta. TACI-Ig or atacicept is still under study. And there are also other things that try and tolerize the immune system. So the definition of tolerance has to do with you can make a cell not react to something, but if you take it out of its conditions where you're making it not react, it can recover reactivity. Because otherwise there's a question on whether or not it's just on its way to dying. But you should be able to, if you take it out of if you have it in conditions that make it tolerant. So imagine that you have, you're in a room with 6 kids at the holiday time. And if you're not feeling well, you're not going to tolerate that situation very well. If you go into a quiet room, your tolerance is restored. And so it's the same thing, taking the cell out of their environment. Can it then be responsible to either the same stimuli or other stimuli? Same concept. This is B cells, which most people know about in lupus because they produce antibodies. And so this is where the B cell inhibitors work, like belimumab. In this stage, before you get to plasma cells that produce antibodies. And this stage takes quite a while. And so you wouldn't expect the cell, the anti-growth factor to work right away, because even though your B cells are constantly turning over, it takes several weeks. And then there are other antibodies that deplete B cells that I'll show you on the next slide. Mature B cells do express CD40, which is another target, another protein that's a target for therapy. So this is antibody to belimumab or soluble BLyS. And so you see the antibody hooking onto the molecules of the B cell growth factor that are floating around. And there are other molecules that were antibodies against this receptor that aren't yet in trials or are not yet, have not yet been in successful trials, I should say. So we're actually looking at tofacitinib. So a common theme is with autoimmune diseases, we often repurpose drugs, right? We go for one indication that we think is going to be the fastest route, the most effective in that disease, combined with a fast route through the FDA. And so tofacitinib was approved several years ago for treatment of rheumatoid arthritis, and Hermine Brunner in Cincinnati, whom many of you may have heard speak, wrote a trial that Pfizer is the original sponsor of, and we're a sub-site for her. And we're looking at patients who have systemic lupus erythematosus and skin lupus. They have to have a disease activity. The skin lupus disease activity score most commonly used is called CLASI, cutaneous lupus activity score. And so it's got to be moderately active or have a CLASI of 8. And you have to have underlying systemic lupus. So if you just have discoid lupus, while this may eventually be proven to be an effective agent, this is not the population we're studying. So I want to switch gears a little and talk about mesenchymal stem cells. And I'm going to talk a little bit about my rheumatoid arthritis program, because I think of that program as a template for how we might go forth with other programs and other diseases. And we're trying to I hold an investigational new drug, an IND for this substance, which means that the FDA closely monitors our trial. And then the next target is systemic onset juvenile arthritis, which in some ways shares some features with lupus. And I think we're waiting on Gary Gilkison's trial, I think, before moving into other kinds of applications for this. Gary Gilkison is at the Medical College of South Carolina, and he's got an umbilical cord product that's a mesenchymal stem cell. And they got funding from NIH, and I think Lupus Foundation heavily subsidized some of that trial. And so we're all sort of awaiting those results. Gary was nice enough to have me look at the trial, and we're an alternate site, but all the other sites signed on. So if they lag in recruitment, we could bring this to Cleveland. So mesenchymal stem cells were initially discovered in bone marrow. Arnie Kaplan, who I collaborate with, was one of the original people, and they were really named for their ability to regenerate cartilage, bone, muscle, fat, and skin. And so Arnold Kaplan's a skeletal biologist, and what he thought is, well, if you get a defect in osteoarthritis, you can put mesenchymal stem cells in and fix that defect. Unfortunately, that's a failed concept because they do make cartilage, but they don't make cartilage that is strong enough to weight bear. So we have ear cartilage, but we probably don't want to run a marathon on our ear cartilage or fibrocartilage. But we could, if we're young and healthy, run a marathon potentially on our knees, on our knee cartilage. So all MSCs and he likes to call them medicinal signaling cells now, so I'm going to have to change that sign, and I'll show you why. And they're around blood vessels, or they're pericytes, and they can be found in fat, gum tissue, umbilical cord, as well as in bone marrow and a number of others. So this was a diagram that we revised and published in 2011. And so you can see that the MSCs, and I'm just going to use MSCs because I don't want to have a big argument with Arnold about what they should be called. And they can go into bone, cartilage, muscle, marrow, tendon, ligament, and connective tissue disease. But really they can do it in a petri dish in the lab. And so that was really the first observation and first hope that it would restore that. And Arnold will tell you that his ideas for 30 years doing this were all wrong. And the most value of this MSCs is the ability to go to sites that are injured or have inflammation and tune it down. And so we began a rheumatoid arthritis program. And rheumatoid arthritis, like lupus, is a disabling systemic disease. When I graduated fellowship in '94, the 10-year disability rate, work disability rate, was 50%. And rheumatoid arthritis typically has deforming arthritis, as you can see on these x-rays. These don't look like normal joints with a space in between. But we know that lupus can have non-deforming and deforming arthritis also, which was really not recognized until the past 10 years. It's a disease that affects women more than men, which is true for lupus too. Both remain a serious cause of morbidity and mortality. And even rheumatoid arthritis is heterogeneous. So some people have antibodies against themselves, rheumatoid factor, or anti-protein peptide citrinolylated antibodies, or APCA, or CCP. But some people don't. And so it's probably it's at least 2 diseases most likely, and maybe more. Okay. And so we know that, at least in lupus, that there are environmental factors, and probably in RA and also in lupus. So if you have bad gum disease or if you have lung disease, you rank up an enzyme called PADI4, and that takes an amino acid called arginine to be a citrulline. And citrulline are found in bacteria, but not in us. And so rheumatoid arthritis patients make autoantibodies against citrulline that then cross-react in the joint. And we know that there are environmental triggers also in lupus, that the gut microbiome is likely to be important in both rheumatoid arthritis and lupus. So we decided that there was a need to have different therapeutics in rheumatoid arthritis. As you know, the field of therapeutics is pretty crowded. My husband was teasing me last night as I was finishing up the talk, and we were watching TV from 10 to 11, and he said, gosh, this is the first non-rheumatoid arthritis commercial, and what was it a commercial for? It was a commercial for denosumab or Prolia, right? So I think we saw 6 within the hour. And so what we know is, like lupus patients, patients with seropositive rheumatoid arthritis rarely get completely off medicines. Less than 5% get completely off any medicine. And there are goals both to alleviate symptoms and to halt damage. So I think that's really a commonality. And we all want to try and induce the immunologic tolerance, which I was talking about, and really return the immune system to pre-RA state, though I could probably put a stop sign there and say lupus too. And disease so the disease-modifying anti-rheumatic drugs, some are shared with rheumatoid arthritis and some are really unique to lupus. And biological ones are expensive. They're effective in some, but not all patients in lupus. And traditionally, it's a single target. So why do we want to study MSCs? We think that they go, when you put them in to an IV, that they go to sites of injury and inflammation and help to tune down the immune system. Part of what we react to is molecules on cells that, like B cells or macrophages or dendritic cells. So abatacept, which is used in RA, binds to these. So these cells express very low levels of molecules that the immune system is going to react to, transplant antigens, for example. And that's why we can give people stem cells that aren't MSCs that are not their own. And they're largely invisible to the recipient immune system. So you've all heard about people who get bone marrow transplant and have to have a lot of chemotherapy before. That's not what we're talking about with MSCs. And so this is the way they work. Right? So what we don't know is, so we know that for some of the actions on T cells, that they help increase T regulatory cells or make them better regulators, and that they tune down maturation of dendritic cells, which then produce interferon alpha. They appear to calm down B cells here. But there are many mechanisms of action, and you can understand that they may act differently in RA than in lupus. And so we're beginning to think about how do we customize these cells to really respond to the different challenges immunologically in different diseases. And this is Gary Gilkison. I was relieved to see when he published this a couple years later that he also talked about MSCs homing to the site of inflammation, things being used to stimulate T regulatory cells, tune down B cells, and antigen-presenting cells, which are things like dendritic cells and macrophages. And so I think the concept is similar. Now, the FDA has on their website that we like to think about these homing to the site. And so if you have a burn, they're going to repair skin. And they're going to alter themselves, because they're a living cell, to do what's necessary. The FDA has said nobody has presented enough data to make those kinds of claims. So right now it's a hypothesis, not something that's proven. And the other thing about mesenchymal stem cells is we think that in certain situations that they get senescent prematurely. And so we're worried about senescence in autoimmune, in patients with autoimmune disease have MSCs, but the evidence is this much, not this much, in terms of that. So it's important that you understand not only what's known, but what's not known. Okay, so they've been used in graft versus host disease. There's no chemo or preconditioning regimen. So the FDA has objected to the third sentence, which is that they sense and repair local inflammation and damage. But we know that they don't really interfere with host immune systems. So if you get rituximab and have no B cells, you're producing less antibodies against bad things and good things. These we don't really think is suppressing the whole immune system, but perhaps being more targeted. And so when we started the trial in RA, we had the hypothesis that it can induce immune tolerance or do a reset. And we are studying things to try and understand early on if they have a biological effect, because you may not want to give continuous doses eventually, or a second dose to patients who are non-responders, right? So right now we say with most of the lupus drugs, we can only poorly predict who's going to respond to what. But it would be nice to be able to give a day or two of a medicine, tell you whether or not you're going to respond, and if you're not going to respond, go on to the next thing. And so that's really a therapeutic goal, I think, for all of the autoimmune communities. So are they safe? They've been shown to be safe in a myriad of diseases. And we pre-med typically with acetaminophen and Benadryl, Tylenol and Benadryl. So no chemo. No tumor formation has been observed in the trials that are regulated by the FDA, although I did get up one morning about a year and a half ago as we were getting started with this trial, and in the New York Times was a picture of somebody with a spinal cord tumor who got stem cells that weren't regulated in some clinic in Germany, and God only knows what happened. Did they get their own? We think about stem cells, if they're from somebody else, as being hit and run because they're gone from the immune system within 24 to 48 hours. So we think about, if they're not your own stem cells that have been reinfused, that somebody else's comes in, does what it needs to do, and then your own body brings in the cells that end up doing the repair. Like the cartilage that MSCs don't appropriately repair, it's really host factors that are tuning the immune system down, and the mesenchymal stem cells are there to get it started but disappear quickly if they're not your own. Tumor formation is probably a little bit more concerning if it's your own stem cells because they may not disappear. They live in you, but for some reason they aren't able to repair just endogenously. And no MSC-related infections have been observed. Those are some safety references. So this is the trial. That's on clinicaltrials.gov. If anybody has friends or family members that have early rheumatoid arthritis, please direct them towards that clinicaltrials.gov site. And this is not for you to read every inclusion and exclusion criteria, but I want to see what your observations are. So these are the inclusion criteria, and these are the exclusion criteria. So what does that make you think? It's really hard sometimes to find people who fit into clinical trials. And so we're interested in measuring safety in this trial, biologic markers to know whether or not we're having any effect, patient-reported outcomes, because some of what we've seen is that in the literature is that patients may report feeling better after getting mesenchymal stem cells, and the biologic effect may lag. And it's a dose-ranging study because we don't know the ideal dose, and it's unclear that there's a regular dose response. So usually if you give a drug, that the higher you get, the more response you get, and then you get to a point of toxicity. This is not as clear with mesenchymal stem cells. Is there a dose response, or is there an ideal dose? And it really is at higher doses you don't want it, and at lower doses you don't want it. And this is Gary Gilkison's trial. Also on clinicaltrials.gov. This is called MISLE. And again, this is what they're looking at. Their primary outcome value measure is something called the SLE Responder Index, which is a composite measure. So it takes into account things on physical, things, how patients feel, and some lab measurements. And these are the inclusion criteria. And this is the exclusion criteria. So again, it's really hard to get people into trials, and there may be certain segments of the population that volunteer and feel less anxious about it, and certain people who feel more anxious about it. But if we don't study it in a range of people, then we will only know, let's say this was, a white, if we only had white women in the trial who don't reflect necessarily the full spectrum of who lupus affects, then how are you going to know if what worked in white women worked in everybody else? And so sometimes that's a challenge explaining to people, but if we don't study people who the disease affects primarily, we're not going to know if it works in the lion's share of people. And so I'm in what's called the Valley of Death. So, Gary Gilkison's here. He got more than $20 million between NIAID and the Lupus Foundation. My trial is being done on a little more than $400,000, but it's only 20 people. His is much more. But so we validated a target. We had a lead cell that we wanted to use. We're here, but the FDA has told us by the time we get to here, we're going to have to know which donor should go to what person and have a method of deciding. So is the same off-the-shelf product good for every disease? Or do we need to select donors so that there's a match for, there might be one product that's better for RA and one that's better for lupus? And then even within lupus, are there different donors that match different individuals? So should we have 3 products when we do a trial? And we'd like to be able to do some experiment in the laboratory saying if we're giving you some cells, we want to do what we think is the best match for you. And all that is really uncharted territory. And this requires a lot of funding. This is what drug companies do best. This is where things developed by investigators fall off, and that's why it's so important to have other sources than federal sources for funding. There are some non-RA diseases. These are some other. There have been trials in scleroderma and Sjogren's, vasculitis. So what's the best clinical trial design? Still unknown. Whoops, sorry, I didn't mean to shut that off. How do I? Okay. Should we be doing single dose or multiple dose? We asked to do more than one dose in our trial, and the FDA said, your product's never been in this disease, you'll do one dose. Need to develop potency assays to try and reduce variability of the product, customize it to the disease, and then we need to understand as a community, are there biologic measures that we can use early on to know whether or not somebody's responding so that we can stop it? And this really applies to all drug development. Cells from you or cells from other people. To have an off-the-shelf product that's readily available immediately, you'd really need to have an off-the-shelf or allogeneic product from somebody else. There may be certain situations where autologous or your own stem cells are more appropriate. So we did not initially plan to use a placebo. And we were asked to put that in by the FDA, and they said, we won't force you. And at the NIH study section, we got a score that just missed funding, and one of the main criticisms is we didn't have a placebo group. So within the 20 patients we're infusing, 5 patients will get sham infusion, just infusion with normal saline. And they said even if the clinical, you can't distinguish people clinically, if we're looking for this thing at 48 hours or 1 week or 2 weeks or 4 weeks, then those will be real. If there's an effect, it'll be really helpful to have patients who got placebo infusion. Advantage, they might offer hope for cure or appear safe. We talked about hit and run, low infection risk, disadvantages. Early in clinical development, they're expensive to produce right now, and currently it would be difficult to produce them on the same scale as, say, methotrexate, or the same scale even as something like belimumab. None of this gets done in a vacuum. These are partners in this. Athersys, I did not show you the work with Athersys that got me started on some of this. And they're a company in town and somebody to watch. They have some clinical trials going on mostly in stroke and MI. And so in our division, the human clinical trials, I think many of you have met Dr. Ballou. You've now met me. Dr. Antonelli is a more junior faculty member who trained with us, who's excellent, and she has also been involved in some of the clinical trials and evaluating within Lucin some of the things that are brought for vetting in terms of compounds. Julia Bolcomez is she's a pediatric rheumatologist, and she is using human MSCs in lupus mice and looking particularly at nephritis, and I think there'll be a manuscript that hopefully will be formally accepted soon. And that's it.